activation of a particular gene in a DNA would result to the synthesis of a particular protein. Okay, ultimately. But this pass through this process, transcription, which is of course the transfer of genetic information from the DNA to the RNA or messenger RNA. And whatever information is translated into the mRNA, transferred into mRNA, then that will be translated later into protein. Okay? So, I think you have this central dogma of molecular biology. I discussed this already. Now, this is what happened, class, when the genetic material is the DNA. So whatever information coded in the DNA that will be copied into messenger RNA, and of course, that whatever information is coded now in the messenger RNA, that will be translated into protein. However, there are some molecules, like for example, some viruses, whose genetic material is not DNA, but RNA. Like for example, HIV, no? The one that caused AIDS. Now, its genetic material is not DNA but RNA. The same is true with the coronavirus. Its genetic material is not DNA but RNA. The same is true with tobacco mosaic virus or TMV. Its genetic material is RNA. So the RNA is, you have this so-called reverse transcriptase. This is now the enzyme that will convert RNA to DNA. And so on and so forth. Okay? So, you have that special enzyme. What is unique among them is they have this special enzyme called reverse transcriptase that will change the RNA into DNA. Okay? So you have that transcription. Now, transcription is the process by which information encoded in the DNA molecule is copied into messenger RNA. And who will do the transcription process? Now, if during replication process we have DNA polymerases, here in the transcription process we have RNA polymerases. Are you with me? Now, what is unique in RNA polymerases is that, so they are the one responsible for polymerization. One nucleotide, another nucleotide, joined together. So that's the role of RNA polymerases for the formation of the whole RNA molecule. Now, what is unique in RNA polymerases is that they can synthesize, they can start or do the starting of polymerization. They don't need any primer anymore. So, they can start the process right away. So, as long as they already have the DNA template, then you have now the transcription process will immediately proceed. So unlike the DNA polymerases, RNA polymerases could start the process of polymerization. Okay? Now, for eukaryotic cells like us, we have 3 types of RNA polymerases. So we have RNA polymerases 1, 2, and 3. The role: the RNA polymerase 1 is responsible for the formation or the synthesis of rRNA or ribosomal RNA. So kinsa maghimo sa ribosomal RNA is the RNA polymerase 1. How about the RNA polymerase 2? It is responsible for the formation of mRNA. And for the RNA polymerase 3, it's responsible for the formation of tRNA and the 5' sRNA. rRNA. So those are the different assignments for these different RNA polymerases. That is for eukaryotic. But for prokaryotic cells like bacteria, they only have single RNA polymerase that will do polymerization. But this RNA polymerase, the single RNA polymerase, have 5 subunits. They have 5 subunits. Now, what are these different subunits? So there are 2 alpha subunits. It's there in our group chat. The 2 alpha subunits, the beta subunit, beta prime, and the sigma subunit. And they have their different assignments, their different role. Now, for the 2 alpha subunits, the role is for enzyme assembly promoter recognition. So, who will recognize the promoter that will do the assembling of the enzymes? You know what? During the process, all enzymes must be assembled. No? So, it's done by promoter. Oy, dali na mo. Helicase, polymerase, mga different subunits. Helicase, isomerases, dali na. Diareses, dali na. Mag-start na ta. No? So, assembling of enzymes is done by a promoter. No? And the one that recognizes the promoter for the enzyme assembly is the 2 alpha subunits. Now, for the beta subunit, is responsible for, this is actually the catalytic center. This is the center for the catalysis of the enzymes. No? So, where is catalysis occur? We say catalysis, it's done by catalysts or mga enzymes that speed up the reactions. No? So, it's done in the beta subunit. And for the beta prime subunit, this is where the catalytic center template binding of. So, you have now the DNA template. So, asan ni siya mo bind? This is for the bacteria, prokaryotic. Now, this DNA template will bind in the beta prime subunit of the RNA polymerase. Because this is the one that would serve as the template, the hulmahan, molder, for the new, or say for the formation of mRNA. And for the sigma subunit, this is the one that will do the promotion. Katong tigtawag. Dali na. Paging this enzyme helicase. So, kinsa ang mag-page? Kinsa ang ga-promote? Kani si sigma. Mo ni ang promoter. Okay. Now, transcription follows these 4 stages. We have the promoter recognition, initiation, elongation, and termination. So, it's there in our group chat. I sent it already. Okay. Now, this one, promoter recognition and initial melting. So, again, like DNA replication, there is what we call as melting of the DNA. What's the purpose of melting? For double strand separation. So, dapat ma-separate sila. So, na-fourth na po method, na-fourth. So, exposed na ang mga nitrogenous bases for the possible template. Let's say, for example, after separation mo na ni, ang sequence. Say, TAC, CCG. So, this is one codon, this is another codon, so on and so forth. Okay? For our DNA template. Now, during initiation, of course, there's what we call as ternary complex formation. Anong na ternary man? Ternary complex formation. Okay. Now, you have here, ang kanina, binary. You have binary complex formation, meaning na duha Now this one, ay mo siya later on tulo. Okay? Duha ka strands din plus the new strand of the mRNA at this point. So it starts there. That starts the initiation process. So you have now the enzyme polymerase. So you have here. So it's unwind by helicase. So na-expose na. So this is now the timplet strand. So kani siya, morning timplet strand. Morning kopyahan sa bago nga RNA. So pag-bind ana sa RNA polymerase, okay, it does not require any primer. So immediately, ang RNA polymerase, pag-expose ani sa first nitrogenous base, na-expose na sa DNA template, immediately, the RNA polymerase will bind. Pag-bind ana, polymerization na yan. That initiates. So, that's why there's ternary complex formation na. 1, 2, 3. Ternary complex formation. So on and so forth. That is, after initiation, that is elongation. So, after T, say, kung kani siya ang T, pag-bind ani diri sa RNA polymerase, okay, so, start na dayon ang transcription process. So, okay, kani siya, T man ni siya, so this is A. How about this? This is adenine. So, ang partner ani is anong human? Kay RNA man siya. Okay? So, wala may timing ang RNA. C, G. C, G, C, G, G, C, so on and so forth. That is elongation process. And after that, whatever is the length of the DNA template is also the length of the primary transcript mRNA. Exactly. Kung sa kataas-on ang template, mapod kataas ang primary transcript mRNA. But take note, class, not all of these primary transcript mRNA is said to be coding sequences. They are made up mostly of introns or non-coding sequences. Only a few or around 10% would be exons or coding sequences which can be translated into protein. So only coding sequences will be translated into protein. Okay, now primary transcript mRNA is very, very long. And if, say, because transcription occurs in the nucleus, it cannot get out from the nucleus to the cytoplasm. Ngano mo ato siya cytoplasm? Iya ang pangitaon ang ribosome. Ngano man? Kaya ang ribosome mo site sa protein synthesis, sa translation. So kinahanglan siya mo gawas from the nucleus where replication and transcription occurs. It has to pass through the nucleus, passing through the nuclear pore. So muagi sa siya diri sa nuclear pore or nuclear pores. However, kani kung ang primary transcript mRNA muagi diri, this will be degraded by degrading enzymes called mga nucleases. These are degrading enzymes na kung muagi gani ining primary transcript mRNA, i-degrade niya so ulay ma-translate into protein. So, it has to undergo maturation process first to avoid degradation by these nucleases in the nuclear pore. Are you with me? Are you with me? Okay. So, what is this maturation process? So, I have my version of explaining or say making a summary of this process. Okay, so we have these three processes that would say mature this primary transcript mRNA. So it has to undergo maturation process. First, there is what we call as end capping. Now, end capping is the addition of methyl guanosine at the 5' end of mRNA. Okay, say for example, if this is 3', one yung 5', kani 5', one yung 3'. So, meaning, end capping, dugangan ni muna siya diha o kaluan ni muna siya diha ang area. Are you with me? That is end capping. Say, if nursing students were capped before or prior to clinical exposure. Now, this one, before this mRNA gets out from the nucleus to the cytoplasm for translation, they have to be capped with methyl guanosine. So, kaluan. Nga naman, that is for protection. Kaya kani siya, these are composed of coding sequences na pwede i-degrade by nucleases, mga degrading enzymes in the nuclear pores. Now, another process of maturation of this primary transcript mRNA is the poly-A tailing. Poly-A, poly means daghan. A, addition of adenyl groups. So, butangan ako diri o adenyl group. So, naa siya'y sapatos. Sapatusan po nimo. Are you with me? Why? Because kanina portion po di po siya i-degrade by the degrading enzymes called nucleases as it pass through the nuclear pores. So, naay mga shields. God is our shield. He is our shield. So, even when He constructed or created this, He put shields. So, we will not be degraded by our enemies. Okay. So, nanay, kalo, o sapatos. Naay capping and capping na ako'y poly-A tailing. So, that preserve the important exons here or the coding sequences. And another, the last is the splicing of primary transcript mRNA. When we say splicing, the cutting of those non-coding sequences introns and the joining of all the exons or the coding sequences. Di ba taas man ka itong primary transcript? Katong mga ulay dili ma-translate nga mga introns. Pagtangputlon to siya. Pamutlon na siya. So, ang length now would be about 1/10 of the total length of the primary transcript mRNA after maturation process. Okay, nga naman. That is due to the splicing of the introns or the non-coding sequences and the joining of these exons or the coding sequences. And who will do the joining? It's done by lipase. So after that, the mRNA is already mature that it can pass through now from the nucleus to the cytoplasm. And what is its target organelle? Okay, the ribosomes. The target is the ribosome. Okay? So, any question? In the transcription. Okay, so let's start discussing translation process. Now, while the matured mRNA pass through from the nucleus to the cytoplasm, on the other hand, the ribosome, as part of the initiation process during translation, will be dissociated. So there is what we call as that is part of the initiation process for translation. There is what we call as dissociation of small and large subunit ribosomes. In other words, katong dako o ang gamay na subunits would be separated. Remember, naan sila diri sa cytoplasm. So paggawas While gahulat sila sa matured mRNA na magawas from the nucleus, nag-dissociate na ang ribosome. There is separation of the larger and the smaller subunit. And another part of initiation is the complex formations. So natay initial complex formation. Pag dissociate ato, pag separate na sa small and large subunit, ang small subunit will bind with another proteins. We call that as initiation factors 1 and 3. So there is what we call as binding of this small subunit with initiation factors 1 and 3. So that is initial complex. Are you with me? On the other hand, so pag-bind sa small subunit with IF1 and 3, mo-bind the code for final complex formation ang initiation factor 2 and the tRNA. Here we are, we have now the tRNA. This time, ang baggage sa tRNA is methionine. That is the amino acid, the first amino acid. Methionine is the starting codon. Okay? So, again, after dissociation of this small and large subunit ribosome, there is binding of the small subunit with the IF1 and 3. And after that, there is final complex formation where the IF2 will bind with the initial complex plus the tRNA carrying now the first amino acid, methionine. And that is the final complex. So, nara sila din. Nagulat. Okay. Kanang final complex, paggawas sa matured mRNA, immediately they will bind. So there is what we call as binding of larger subunit to mRNA to form a peptide bond at the met site. So, diri na ta sa translation. No? So you have now the small subunit ribosome plus the IF1, IF3, IF2. Okay? And then plus we have the tRNA with, you have here the first amino acid called methionine. Are you with me? So, naatay methionine. So, pag-bind karon sa final complex with the methionine, meaning protein synthesis starts. This is the starting codon, ang methionine. So that means protein synthesis starts. And immediately, elongation will follow. No? Elongation. So how? Now, on the other hand, katong larger subunit ribosome will bind. Mo-bind na siya diri. Ang larger subunit ribosome. Now, during the binding of the larger subunit, immediately, IF, initiation factors 1, 3, and 2 will be dissociated. It will leave the complex, plus that of the mRNA. Also, the tRNA will leave. And what is left is only methionine. So the binding of the larger subunit causes the dissociation of the initiation factors 1, 2, and 3 plus the tRNA, leaving the first amino acid called methionine. And that is now the starting codon. Now, since there is already methionine here, now another result of the binding of this larger subunit is the formation of the first peptide bond. So, natay first peptide bond. Are you with me? So that is what is accomplished in the binding of the larger subunit. Dissociation, initiation factors 1, 2, and 3, plus the tRNA, and leaving only the first amino acid called methionine, and the formation of the peptide bond. And now, next is the movement of the small subunit again to the next codon. Move na po ang small subunit. Okay? Now, pag move ani sa subunit, mukhang ina po ang tRNA. Carrying, unsa man yung GGC? GGC is GGC is glycine. Carrying the amino acid called glycine. So, naana po tayo second amino acid nga gihatod sa tRNA. And, of course, you have now, paghatod ana sa tRNA with the glycine, mumove na po ang larger subunit na napoy peptide bond formation, so on and so forth. Padayon na din na siya. Until, okay, of course, we have this different binding sites of ribosome. You have the A site. A site is the amino acid site. That is where amino acid is found. So, you have here. So, kani siya, mo ni ang amino acid site. Kani sa ribosome. And you have the P site or the peptidyl site. Kani mo ni ang site sa peptide. Kani mo ni ang site sa peptide bond or the peptidyl site for the have here sa ribosome. Sa ribosome. And you have the E site or the exit site. Exit site where tRNA will get out. After niya naghatod og specific amino acid. So on and so forth. Are you with me?
Yes, ma'am.
Now, until such a time, class, that is we have there 1, 2, 3. Now, sorry. Now, say for example, ang last codon na dili is uwa, any of this, uwa, uwag, uga. Uwag.
I like yan, no?
Now, these are mga termination codons. So, pag-move karon, okay, pag-move karon sa small subunit ribosome, nag-move siya diri. Pag-move karon, pagtanaw sa pagtanaw karon kay UAG or uwa or uwag or uga. Now, this codon, there would be no specific codon for this. Ang wala kay tRNA nga muhatod. It does not code for any amino acid. Wala kay tRNA nga mubind. Ngano man? Because any of this, paggawas aning uwag or uga or uwa, now it codes for water. So H2O. So kanang water maunay mo hydrolyze sa last peptide bond. So meaning diha siya taman. Wala na siya. I-hydrolyze na siya sa water. Okay. So these codons do not code for amino acid, but water to hydrolyze the last peptide and form at the last amino acid. So for example, mga hinig, glycine na ang last amino acid. So, ang katong peptide na na-form after the movement of the larger subunit will be hydrolyzed by water. After coming out of this termination codon or any of these termination codons. Okay, question? Now, you can now read the details in your textbook, no, or say in the PowerPoint. Ha? Pareho na siya. You just understand it. Ang presentation mo, good kanya ako synthesize, summarize.
guys.
Guys, ganit kayo, ma'am. Tayo nice.
Oh, so you guys na gani.
Guys.
Okay? So,